It's really nice to be here. I haven't actually been able to get to one of these summer schools for several years, but it's really nice to have the opportunity to come and talk to you and meet some of you and find out about what interests you and hopefully what might inspire you about plants as well. So I've sort of put together a talk this evening. It's a kind of gentle warm-up tonight. We don't have a tutor session on this evening's lecture. That I hope will show you some of the ways I've followed curiosity during my career. And also hopefully convince you that you don't need to know how to do something to make a career out of it. There are quite a few shifts and changes I'm going to explain as we go through. But it's all driven by a genuine interest in how plants work. And I'm lucky enough to be in an environment that lets me follow that without having to have an output in mind, but just genuinely to figure out how stuff happens. So I'm going to start by acknowledging my research group who are responsible for the work I'm going to show you. In fact, most of these people aren't responsible for the work I'm going to show you because I'm going to show you work over many years, and this is the current research group. But just to put a few faces to names, Ben here was sitting where you are about 5 years ago. He's a PhD student in my lab. He's just finishing. And I'm also going to talk about work done by several of these people, including Humberto here and Bhavani, who's up here. So what I want to tell you about is work that's inspired by my role at Cambridge University Botanic Garden. So as Dan said, I've got the good fortune to be the director there. It means I get to spend a lot of time wandering around a 40-acre site that holds 8,000 plant species. We could play some games here. I mean, 8,000 doesn't sound very much, I think. Anyone have any idea how many plant species there are on the planet? I'm not going to let Richard guess if he's in the audience.
50,000.
Oh, that's not bad. Probably nearer 400,000. We don't know the answer, actually, but about 400,000. So 8,000 is about 2% of that species diversity. That's pretty miserable when you think about it as a percentage. Turn the question around, how many species are there in the UK of plant? Yeah?
10,000?
Yeah, no. 2,300-ish. So actually my 8,000 on 40 acres is a pretty impressive biodiversity hotspot. It's about the same number of species that you'd find, a bit less than Madagascar, actually. It's about 9,000, but Madagascar's bigger than 40 acres. So it's a very diverse area. Botanic gardens are amazing things, is the point I'm trying to get across here. If you happen to be near one, go have a look, get to know the diversity of plants. What happens for me is I walk around it supposedly doing my job and thinking about budgets and people and all of those horrible things, and then find plants that interest me and go, oh, how does that work? And then that inspires research directions. And the primary thing I'm interested in is this, the relationship between flowers and the animals that pollinate them, and in particular about how plants modify and evolve their developmental programs that generate flowers so that they generate that enormous diversity of flowers you see if you look across the plant kingdom. So I'm going to talk today about two main research topics that I've worked on over the last few years, but I'm going to show you them from a three-part approach. So my background is actually in developmental biology, so you're going to do some great development practicals this week. You'll hear a lot in many of the other lectures about genes and gene regulatory networks and developmental programs, and that's where I began my research too. But I'm going to show you through the talk that despite the fact that this is what my training is, an awful lot of the work I do is now understanding the function of how flowers attract pollinators and work with them, and about the evolution of the whole system. So I think of this down here as the how question, how do you build the trait? And this over here is the why question, why is it there, what's it for? And if you understand both of those two things, you can look in an evolutionary context and say, so on this phylogenetic tree, this trait is changing here and here. It doesn't matter what the trait is, just these are the points of change. Well, if I know how it's built, I can ask what's happened in the genome or the developmental programs that's allowed that change. And if I know what it's for and how it functions, I can ask what happens in the adaptive landscape of that plant that's allowed the change. And so by putting the how and why questions together, I can get a picture of how actually you generate that enormous diversity, those 400,000 different species with their different types of flowers. So the first trait I'm going to talk about today is a trait I've been working on since my PhD, and it's the shape of the cells on the epidermis of the petal. So you're looking down a scanning electron microscope at the skin of a petal of an antirrhinum, a snapdragon, and the cells are hexagonally based, roughly, but the key thing I want you to see is that they're conical. They're pointing out of the screen towards you. They're like little pyramids. And that's unusual. That's not what cells look like on a leaf or a stem or any other part of a plant, but it's very, very common for flowers. most petals have cone-shaped conical cells on their epidermis. And so my PhD was about figuring out how those were built. And I did that in Antirrhinum, in the garden snapdragon, where we had identified a mutant that didn't make those conical cells. So I'm going to try and hopefully be able to show you this. This is a wild-type snapdragon. It has conical cells on its petal. And the reason I know it has conical cells is that I can see the line where they stop. So I hope you can see that there's a line there, and above that line it's darker, deeper coloured pink, and below it it's paler. This is a mutant which doesn't have the conical cells, and it's all the same colour as the paler part here. And then this is an unstable mutant, which has the conical cells and flat ones. You can see them in close-up, and the conical celled regions are the dark patches, and the pale tissue is the flat cells. So those conical cells are enhancing flower colour. And that's actually quite easy to explain from a physical point of view. They act as little lenses. And they focus the light into the centre of the cell where the pigment is in the vacuole, and that means more white light's absorbed, and it dilutes the amount of white that's reflected off, and the flower looks deeper in colour. So we'd identified this mutant. My job was to clone the gene, and to cut a very long story short, those of you who've gone to do PhDs will know just how long a PhD can feel, even if it is only 3 or 4 years. The gene turns out to, we called it mixture, and it encodes a transcription factor from this family called the MIB family. So these are master regulators that turn on developmental pathways. And what it does is regulate cell growth in the Z plane. So that's the plane towards you as you're looking at this slide. So here are conical cells induced by mixture sitting in a sea of flat cells where the mixture gene is mutated. And the reason we have this unstable phenotype is that this mutation was caused by a transposon, a jumping gene that was inserted in the genome. When it jumps out, you get conical cells. When it stays in, you get flat cells. And we use the transposon to clone it. So that was my training in science. Nice plant, pretty flower, but actually molecular biology in the lab, gels, DNA extractions, clone this gene. And yet every time I gave a talk, and any conference I ever went to, the only thing anybody ever asked me as the first question was, what are those conical cells for? Why does the petal have them? Is that change in colour really important or significant? And so this is where I started thinking about function and wanting to extend out of just developmental biology into this more broad way of looking at plants. And so I designed an experiment towards the end of my PhD, which was very simple, to try and actually get at this question. And what I wanted to know was, well, okay, what did bees think? You can see this is a PhD drawing of a bee. It's got no legs at all and the wrong number of wings, I learned. So I have my wild-type flower with its conical cells. I had my flat-celled mutant. And I also made a cross between the flat-celled mutant and a white mutant that doesn't make pigment. So now I have a white flower with conical cells and a white flower with flat cells. And the idea was, well, I'll put all these out in a field, in a randomized trial, and I'll see what the bees think of them, and I'll see whether actually those conical cells make any difference at all to attracting bees. And the experiment was very simple. So we grew up these plants, we emasculated the flowers, so opened them when they were still at the bud stage and pulled out the anthers and then closed them back up and tagged them. Now that meant that an emasculated flower couldn't self-pollinate. It would not make a seed capsule unless a bee came into it with pollen. And so we could essentially go back a few weeks later and count how many of the flowers that we tagged and therefore had emasculated had set seed. And we said if it set seed, it's been visited by a bee, and if it doesn't have any seed, it's not been visited by a bee. And we can compare the numbers for the 4 different varieties. I learned a lot about fieldwork in this experiment. If you do fieldwork ever, you'll learn that it's never very simple. There's a very large difference in numbers of bees in my field over the 2 years I did this experiment. As you can see, that's because it rained perpetually for the first year and not for the second. These things happen with fieldwork. But the pattern is clear. Conical-celled flowers, more of them are setting seed in both years than the flat-celled flowers. So those conical cells do seem to enhance pollinator attraction. They make the flowers more attractive to bees. So that was cool. And we assumed it was because of that lensing effect I showed you. Except when you look at the data for the white flowers, where there's no pigment, no visible difference, no lensing effect, you get the same pattern. The conical-celled flowers attract more pollinators than the flat-celled ones in both years. Red flowers attract more bees than white flowers. That's quite nice data as well. So now we had a confusing result to explain. We thought we understood what conical cells were for. They certainly attract bees, but not through the visible effect that we can see because there's no pigment to be enhanced in these white flowers. So once I'd moved to Cambridge and I set up my own research group, I wanted to come back to this question and try and understand it in more detail. And so I set up a collaboration with Lars Chitka at Queen Mary University in London. He's a bee cognition expert, and developed a bee behaviour lab that I could use inside, so get away from this rain some years and sun some years problem, and actually get more robust data on what was going on. So I like to say this is the lowest tech experimental setup in any research lab in Cambridge. I'm quite proud of that. It's a plywood box with a perspex lid. We call it a flight arena because it sounds better. And it's connected by this plastic tube to a box of bumblebees. So this is Bombus terrestris, the buff-tailed bumblebee. We buy them from suppliers who supply them to people growing tomatoes or strawberries in polytunnels who need to bring in bees to pollinate their crops. So when the bees come to us, we have a queen and some larvae in here. They've never been out of the box. The idea is the larvae hatch into workers. The workers go through the tube, which we can gate by putting bits of cardboard in it. And they fly around in the flight arena looking for food, which they then take back through the tube to the colony and feed to the larvae and queen. More larvae hatch and so on. And so while the bees are out foraging in our flight arena, we train them. And when they come to us, they've never met a flower, so we can tell them anything we like is a flower. And this is our standard experimental flower. It's a test tube with the lid painted pink. There's a little cup here on the top. It's an Eppendorf cap, and it's got sugar solution in it. So the bees are quite happy with this as a fake flower. They collect the sugar, drink it, they take it back to the colony and feed it to the larvae. And while they're out flying in the flight arena, we can open the little gates on the side and we can pop a bit of paint. I hope you can see a blue patch here, a red patch here on their backs, or a sticker. And that means we can actually get really good, robust data that you can do statistics with. So if you do these sorts of experiments in the field, you don't know whether you're looking at the same bee you saw 5 minutes ago or a different bee. With this setup, I can say I'd like to know what the first 100 choices that 20 individual bees make are, for instance, and we can collect that data because we can follow individual bees. So I'm going to cut again a long story short and tell you that the answer to the question about what those conical cells are for turned out to not be to do with colour at all, but to do with the way bees handle snapdragon flowers. So if you look at this bee on this snapdragon flower, what she's actually doing is sitting on an entirely vertical surface, and she's doing that because her claws here are locked into those cells like little the bumps on a climbing wall. They're providing her with grip so that she can handle that flower more successfully. And we showed this after a lot of experiments looking at colour and temperature and scent and all the other things we thought might be happening that weren't, with a very simple experiment in the flight arena. So here we have snapdragon flowers, and we used our wild type with the conical cells and our mutant with the flat cells, and we gave them to the bees this way up. So here's a little, it's actually a pipette tip with sugar solution in it. All the bee has to do is land on this horizontal surface and stick its tongue down here to drink. And we also gave them the normal way round that a bee would meet a snapdragon flower, where it has to land vertically and then feed down to the tube. And we said, what's your preference for the conical celled flowers in this setup? And in the horizontal setup, where the bee has to land on this nice straightforward platform, there's no preference. The preference for conical cells is 50-50. But when we ask them to land on the flower this way round, then we see nearly a 70% preference for the conical celled flowers. So they only show the preference when it's harder to land. And we can get the same data if we actually make artificial flowers. So here's an experimental design where we've basically made lollipops with conical cells on their surface or flat cells on their surface. And again, we've got two different colour combinations here, don't worry about that. But if we offer these lollipops horizontally, so the bee has to land on top of it, and say, what's your preference for conical celled ones? 50%, just over 50%, depending how we colour code it. If we present them this way, like actual lollipops, so the bee has to land on that vertical surface, we now get a 60% preference for the conical celled form. So those cells are about grip. They're about how the bee hangs onto the flower and handles it. A completely surprising result, because I started off with this mutant that had a colour effect, thinking it was all about colour and the visual effect. And also a surprising result that led us further down the path of trying to figure out how flowers work for bees, even though we started off doing developmental biology. Because we wondered, well, actually, if conical cells make such a difference, are bees able to take that into account in their foraging preferences? And we designed an experiment to test that, to figure out whether it's just a yes, no, I like conical cells, I don't like flat cells, or whether there's actually something more complicated going on there, where the energy spent trying to handle a flower is traded off against the energy you get from the flower. So this is an experiment designed by my postdoc, Hamish, and what he did was produce those artificial flowers, like I showed you, all with conical cells on the surface, so all easy to grip, presented either vertically or horizontally, and he set up 3 different experiments. So different bees did experiment 1, 2, or 3. So one group of bees did an experiment where both the vertical flowers and the horizontal flowers had 35% sucrose solution as the reward on them, so no difference. And he asked them, which do you prefer? A second group of bees had 50% sucrose solution on the vertical flowers, 20% on the horizontal flowers, a big difference. Does that make them prefer the harder ones to manage? And then a third group of bees had a smaller difference, 50% and 35%. So if we do that experiment, I'll show you, nice feedback. Here's a bee feeding on one of those vertical towers. And because the surface has got conical cells, the bee is perfectly happy. She's gripped, she's comfortable, she's able to manage that drop of nectar without any problem. So all the bees did that experiment, and then we were very mean to them. So we replaced the discs with identical setup, but this time with smooth surface, so no conical cells on either the vertical or the horizontal. And so the bees who had learnt that there was no difference were now shown the same experiment with no difference but with smooth surfaces. The bees who'd learnt that there was a big difference were shown the same experiment, big difference but smooth. And the bees that had learned a small difference, again, got the same difference but with smooth surfaces. And smooth surfaces are much harder to handle. So sorry about the noise, but watch the bee trying to feed now from this drop of sucrose on a smooth surfaced disc. She can't grip, her legs are scrabbling, the wings keep beating, and it's really very inefficient. Okay, so the question then was, do they change the preference they've learned when they discover that it's hard to handle? Are they taking into account the energetic efficiency of the feed relative to the energetic efficiency of the reward? So here's the result from the first set of bees. So we've got rough discs, plenty of grip, no difference in the sugar concentration between the vertical and the horizontal, and the bees say, we largely don't care. So this is the proportion of visits that are to the vertical ones. They might slightly prefer the horizontal ones, but essentially they're not bothered either way. And why should they be? They can grip them, and there's the same sugar on them both. Okay. Then we'll look over here at the big difference. So 50% sugar on the vertical, 20% on the horizontal, easy to grip. The bees are almost 100% for the vertical discs with the higher sugar on them. They're sweeter, they're getting more energy from them. And if we look at the middle one, where the difference was less strong, well, it's a slower learning curve, it's not such a strong preference, but again, they're showing us that they're able to work out that those vertical discs, even though they're harder to handle, have got the better sugar in them. So that's what they learned when it was rough and they could get a grip. So what happens when we make them smooth? Well, down here, you might predict what happens when we make them smooth. So they've both got the same sugar in them, but the vertical one's hard to hang on to. What do you think is going to happen? They're all going to go horizontal. They all go horizontal, all right? So a sensible bee making a sensible decision, it's hard to handle those vertical flowers and the sugar reward's the same on both. Why should I care? Over here, there's a big difference. The vertical discs are a lot sweeter, even though they're harder to handle. What did they do? They stuck with it. They worked out that the energetic reward was worth it. Or they don't have the flexibility to change, one or the other. The answer to that comes from the middle experiment. What happens here? If they haven't got the flexibility to change, you'd expect it to stay the same. If they're actually doing a trade-off, you'd expect it to do this. And what happens is that they go to the horizontal discs. So they do the trade-off. In the middle experiment, the bees have worked out the energetic costs of the foraging are not worth the energetic gain because of the low sugar difference. And so using experiments like this, which are absolutely nothing to do with anything I was ever trained to do, has helped us work out how sugar concentration in nectar feeds into bee foraging decisions, how their brains work, how they trade off things in making those decisions, and in combination with an understanding of how viscosity of nectar affects how quickly they can regurgitate it to the offspring, we've actually been able to design optimal nectar concentrations for farmers and crop breeders so that we can try and maximise crops that produce nectar to have the best nectar reward for the wild pollinator populations in those areas to support them. So it's completely different sort of work from anything I thought I would be doing. Now that was the functional stuff. I want to say a bit about the evolution as well. So I said that I like to look at the development, the function and the evolution. So I've just shown you that these conical cells are great. They make it easier for bees to handle flowers. It makes it easier for them to forage. They spend less energy doing so. So a question is, well, do all flowers have them? How old are they? Did the original flowers have them? Have they evolved multiple times? And to answer that, we went back to the base of the angiosperm phylogenetic tree. So if you build a phylogeny of the flowering plants, down at the base there are 3 orders here, and they're known as the Ana grade, the Amberolales, the Nymphales, the Austrobaleales. Amberolales are represented by a single living species, which has really weird flowers, so we ignored that one. The Nymphales, though, are the water lilies, so quite easy to get hold of those and have a look at what they're doing. And here's Kabomba, a little model water lily we work with. It has 6, we call them tepals, because they're neither obviously petals or sepals around the outside, and those tepals have these wonderful, really steep conical cells on them. So it appears that these conical cells are present, at least, in some of the earliest diverging angiosperm orders. Are they made through the same developmental pathway, which would suggest a single origin? Well, we cloned Mixter from Kabomba, and we looked at its expression pattern using quantitative RT-PCR, and the key point here is the big spike here, which is very high expression in the tepal at the stage where the conical cells are about to start forming. Good correlative evidence. It looks like the gene is active in the right place and at the right time to make those cells. We can't make mutants in Kabomba, we can't knock it out, but what we can do is take the gene and put it into another system and see if it can induce conical cells where there aren't any. And so we did that in tobacco, which normally has flat cells on its ovary wall. When we add the Kabomba mixture, we get massive outgrowth, conical cells, trichomes and hairs. So it looks like we have an ancestral situation where this developmental pathway is present at the base of the angiosperms, is able to induce conical cell formation in the petals, and those conical cells make it easier for pollinators to grip the flowers and forage. Great. Except I began by telling you that most flowers have them, but they don't all. So there's another exciting evolutionary question I didn't expect to be answering, but why don't all flowers have conical petal cells? If they're ancient and they're brilliant, why have some flowers lost them? That doesn't make sense. And again, this is where access to a living collection or a botanic garden or a herbarium, which is a collection of dried pressed plants, can come in really handy, because you can actually start to explore the diversity of species and work out whether there are any patterns present. So I'm not going to show you the data, but for instance, we've been working in Nicotiana here, where there are repeated losses of the conical cell form, each time the genus shifts to pollination by moths, as this species is. Moths, of course, don't land on a flower, they hover in front of it, and so they don't need grip. Conical cells are irrelevant. Similarly, this is Rhodochiton, this is a close relative of Antirrhinum, and it's pollinated by hummingbirds, which also don't grip, and even if they did, have much bigger feet. It's lost its conical cells, and again, that's presumably because there's no benefit to the animal that pollinates it. To show you how we do this work in more detail, I'll show you an example from Solanum. So this is Solanum dulcamara, woody nightshade, and the Solanum genus, so this is tomato, potato and so on, is pollinated in a slightly unusual way. So it's bee pollinated, but the bees aren't foraging for nectar, they're foraging for pollen, and the pollen is held in these big yellow anthers, and they have pores, which I hope you can see at the tips. And what the bee does is vibrate the anthers, and pollen flies out and lands on its body, and it grooms that pollen off to feed to the larvae. It's called buzz pollination. So we had a look across the genus Solanum. It's a big genus, and each of these arrows points to an independent loss of the conical cell form within the genus. So it's repeatedly lost, which is very unexpected and unusual. To show you in more detail how we work that out, that is a subset of that. It's one of the clades of that big genus I just showed you, with each of the species marked on it. And every one of the white boxes is a species that we've had a look at the flowers on, and the cells are conical. Every one of the black boxes is a species we've had a look at the flowers, and the cells are flat. And all of the ones that don't have a box, we haven't managed to get hold of the flower yet. But from that, you can work out what the most likely ancestral point at each node in the phylogenetic tree is. And you can work out that there have been at least 1, 2, 3, 4, 5 independent losses of conical cells just in that little section. And then, if we go in detail and look at pairwise combinations of conical and flat-celled species side by side, so here are a couple of pairs, flat cell and conical, flat cell and conical. Again, clone those genes, the mixture genes, and have a look at their expression patterns. This again is quantitative PCR, and the key point is the young bud graph here. The pale bar is the conical-celled species of the pear, the dark bar is the flat-celled species, and basically, if you've got conical cells, you've got more expression of those genes that we know are involved in making them. So we can have a look at how, at the developmental genetic level, this repeated evolution is happening. And we can also look at the functional stuff. My movie doesn't want to play, but I can show you that the bee here is foraging on an artificial flower that we've created with conical cells, with a real Solanum anthocone poking through the middle of it in a hole. Doesn't actually put her feet on the petals at all. If you look, you can see they're embedded in the anthers. And so when they're foraging for pollen in this way, they don't actually grip those petals, they grip the anthers, which explains why the conical cells can afford to be lost. So I've shown you one example, I said I was going to talk about two, of a project that's taken this three-pronged approach, starting from the developmental biology, which I told you very little about, but which is where I started, moving through the function and then into the evolution to try and understand this diversity of different flowering forms and how they work given an ancestral situation and an ancestral developmental pathway. So I'm going to change tack and tell you a similar story about a different project, but this one I'll focus more on the developmental biology because that's what we're mostly doing at the moment, a little bit of function, and end by telling you what the evolutionary work we want to do is. We haven't started it yet. And this is a project that actually stems from this first project I've told you about. So we were working on this conical celled form, and we wanted to try and understand why flowers don't always have an entire petal covered in conical cells, why there might be patterns within the petal. So if you take the whole petal and look down the microscope, why you might see regions that have conical cells and regions that don't. And to explore that, my postdoc, Heather Whitney, who's now at the University of Bristol, was going through the Botanic Garden, picking flowers and putting them under a microscope to have a look. And one day, she came into my office with this flower in her hand, picked in the Botanic Garden in Cambridge, and said, "Okay, I'm going to put it under the microscope to look for conical cells, but how does it make this weird kind of shimmery, bluey, yellow-green thing in these two lower petals?" And in fact, they're not in the two lower petals, it's angle dependent. So if you move that flower around, the shimmery, bluey, greeny stuff would move up here. It depends which angle you're looking at it from. It's a bit like petrol on a puddle. And I looked at the flower and I said, I've got no idea. And we realised that nobody had any idea, that nobody had ever really noticed before, that sometimes flowers could make angle-dependent colour or iridescence, and we had no idea how it worked. So normally when you think about iridescent colour, you think about something like a soap bubble, or as I said, oil on a puddle. It's this changing colour, and as you move your head or the soap bubble moves, the colour that you see changes. And you've probably never thought about that iridescence before, but actually it's really hard to imagine how it's built. So colour in nature normally comes from pigment. The carpet here that I'm standing on is red because there's a pigment in it that's absorbing blue and yellow and green light, and all you're seeing is what's left, which is red. The Gatsby t-shirts are blue because they're absorbing red and yellow and green, and all you're seeing is the blue that's left. And that's how almost all colour in nature works, including almost all flower colour. You can't generate angle-dependent iridescent colour that way. You can only generate colour that looks the same from all angles using a pigment. To make iridescence, you have to use what we call structural colour. And structural colour is when you take a transparent material, so no intrinsic colour, and you structure it in such a way on the nanoscale that it interferes with light, and it reflects some angles at some wavelengths at some angles and other wavelengths at other angles, or it only reflects certain wavelengths. And this can only be done by structuring, which is why it's called structural colour. So there are common examples of structural colour in animals. It's never very common, you know, my cats don't do it, but you've probably seen some of these before. So the rosemary beetle, the red and green stripes here are structural colour, blue on the fish, this blue on the spider. A lot of bird feathers, think about a mallard duck in the sunshine, the green on its neck. But the most common example that people give is the morpho butterfly here, which has a brown pigment, you can see, it's melanin, the same stuff that you make when you have suntan, but no blue pigment. And that blue is produced by a nanoscale structure that reflects only blue light and only a very narrow wavelength band of blue light, which is why it's such an intense colour. So structural colour was known in animals when we first looked at this strange flower, but not in plants. We started to look to see if it was more common, were there actually iridescent flowers that we just never really thought about, and we found quite a few. So I'll show you a few examples. This is a tulip variety called Queen of the Night. There's a purple pigment, but I hope you can see the yellowy, greeny, bluey, iridescent colour on top of that. Here's a daisy doing the same thing as the hibiscus I began with. We've got angle-dependent blue here at the base. But actually, many of the flowers we work on that are iridescent don't look it. So here's a classic example. This is a South African species called Griellum. To me, it just looks intensely, brightly, metallically yellow. I see no angle-dependent colour change. But if I peel its epidermis off, put it on a microscope slide and change the angle you're looking at it, it reflects blue, yellow, red at different angles. So it is an iridescent structure. It's just one that our eyes don't see very well because the yellow is saturating. Of course, it doesn't really matter what our eyes see. It matters what the pollinator's eye sees, and I'll show you how this affects the pollinator in a minute. Now, the good news is that even though we can't always see it, we can measure it. And so we have to be able to do that to say whether a flower is iridescent or not, because, as I say, they're not aiming at our eyes, and sometimes we can't tell. And the way we do that is to measure the light reflected from the petal. So if you look on the left, I think this is the biologist's way of viewing it, and this is the physicist's way. I have to work with physicists for these projects, so I have to put up with their ways of presenting data. So the biologist's way of presenting it, we've got wavelength of light reflected on the x-axis, we've got amount of it, proportion of it on the y-axis, and we've got 7 different curves here. So the purple curve is a flower that is predominantly reflecting in the blue UV part of the spectrum. That flower would look blue or UV blue to us. The yellow curve is a flower that's predominantly reflecting in the yellow part of the spectrum. Here, it would look yellow to us. But actually, all of these curves are taken from the same flower. It's the tulip I showed you earlier. And what we've done is vary the angle at which we're collecting it. So we always shine the white light from the same angle, and then we collect at this angle and that angle and that angle, and we get different wavelength peaks of reflectivity off. And so that's how I can tell it's iridescent. The physicist's way of viewing it is to do the same thing, but we've got wavelength along the x-axis, we've got angle on the y-axis, and we've got a heat map. So the closer to red you are, the more intense the reflection is. If the flower you were looking at was blue, you would have a vertical peak here. It would always reflect at 400 nanometers, whatever angle you looked at it from. The fact that it's a diagonal is what tells you it's iridescent. So at 20 degrees it's blue, and at 10 degrees it's yellow. In fact, that's not a flower, because that's really impressive, perfect iridescence. That's a CD. If you've ever looked at a CD and seen the angles it produces, that's what a flower looks like. So much messier, smudgier. But the diagonal effect here is the key that tells us it's iridescent. We've also got this scattering of light around it caused because it's messier. And I'll come back to that later in the talk. It's quite important. So I compared what we were looking at to what you'd see if you looked at a CD, and actually the flowers are making this iridescence in the same way that a CD does. So if you take a CD and you put it on an electron microscope, these are the data grooves, okay? And the data grooves are spaced about a micron apart, which is the same as a wavelength of light. And as a result, they act as what we call a diffraction grating. So when white light arrives at a diffraction grating, its different components of it are reflected at different angles, so all the blue light sent off in this direction, all the red light sent off in that direction, and what you see is this rainbow effect here on your CD. If you look at the tulip I showed you earlier, on the same scale, same magnification, its surface has these long ridges and grooves on it, again spaced at about a micron apart, also acting as a diffraction grating and generating colour, not as impressively as the CD, but in the same way. Now, the reason it's not as impressive, the reason for all that smudgy stuff on the graph I showed you and for the lack of the full rainbow effect here, is obvious if you look in cross-section. So if you looked at the CD in cross-section, it would be absolutely perfectly regular because it's been printed by a machine. If you look at the tulip in cross-section, we've got a narrow gap here and a bigger gap here. It's a bit irregular, and that's because it's a living thing, okay? So it doesn't produce that exactly perfect degree of iridescence. And this has been one of the most fun parts, I think, of my work over the last 10 years. When I started doing this and first took my hibiscus flower to a colleague in physics and a picture of what it looked like down the microscope and said, could these ridgy things be making the colour? And he went, yeah, that's the diffraction grating. He was fairly sniffy about the quality of this diffraction grating and the messiness of it and the messiness of this and the smudginess of the graphs when he measured them for me. And I spent a lot of time defending the plant and going, well, it's biology, it's never going to be perfect, is it? It's, it's the best you can do with living material, and it's very, it's wet, it's vacuolate, it's full of cells, it's, it's not going to be perfect, but it's not bad. With him going, I'm going to show you through the next few minutes that actually the biology is perfect. You don't need to be a CD. In fact, being a CD would be a bad thing, and that being a flower is better. So starting with the developmental biology, we really wanted to know how the flower built this system of ridges. It's such a cool developmental question. How do you build a structure on your surface at the nanoscale sufficiently regular that it generates colour, and predictably so, these flowers always have it, but on that tiny scale? What's the developmental basis of that? And we spent some time trying to pick out the best flower to work on, because of course Arabidopsis and Lanterinum and wheat and anything else you might think of as a sensible model doesn't have iridescent flowers. And we came back in the end to the hibiscus, which was the very first flower that we'd looked at in the very beginning. It's easy to grow in the lab, it's got a rapid life cycle, it'll actually grow from seed to seed in about 3 months. Small genome, and important at the time, although less so now, it was closely related to several species with sequenced genome. It's in the same family as cotton and cocoa. We have sequenced genome for it now, and so that's not so important. And we began by saying, well, okay, how are we going to study this? One of the first things we worked out was that the region with the ridges is only in the central part where there's also purple pigment. So we've got a sort of purpley red pigment and then the ridges on top of it making the blue colour. So that's quite useful because we can compare what's going on in the white part where the cells are conical and smooth to what's going on in the purple part where they're flat and have the ridges on them. And those ridges, it turns out, when we look in detail, are made of the cuticle. So the cuticle is a bit of the plant that I had never thought of since my undergraduate days, and if I had thought about it, I'd thought was probably very boring. But to remind you what the cuticle is, so here's a cross-section of 2 epidermal cells, cell membrane, cell wall, and then the cuticle is the waxy hydrophobic polymer matrix that the exterior parts of all plants have on them above ground that stops them drying out. Okay, so the point of the cuticle is that when the sun comes out, this water doesn't get sucked out of the cell and the plant doesn't go splat. But our cuticle is arranged weirdly with these ridges on top of it. So we know quite a bit about how cuticles are made, the sort of process from a set of transcription factors called the shine family, which activate the whole thing, the transporters that move the monomers across the membrane, and the enzyme out here, cutin polymerase, that synthesizes cuticle properly out here on the surface. All of that's been well studied in Arabidopsis and also in tomato, because you need it to make your tomato fruit stay alive on the shelf. Without it, tomatoes wouldn't cope after they were picked off the plant. But none of these things have iridescence, and so it didn't help us understand how we got this on the surface rather than just smooth. So the first thing we did as developmental biologists was actually have a look at when and how they develop. So we divided our hibiscus flower development into 5 stages. We looked at them on the microscope, and we discovered that the transition happens between stage 3, when the purple region has smooth square cells, and stage 4 when it has elongated ridge cells. So it's this part in the life cycle where those ridges form. And one of the very first experiments we decided to design was what's called a comparative transcriptome. And you'll come across these a lot in the plant science literature. They're really useful ways of figuring out what's going on with a problem. So the idea was, here's the experimental design, we took stage 3 petals, so that's before the ridges appear, and stage 4 petals, when they're starting to appear, and we took the white region, where there are never going to be ridges, and the purple region, where there are going to be ridges. So we dissected all of these out, and then we extracted RNA, and we sequenced all of the genes that are expressed in each of those 4 tissue samples to see what was there. And so if you do a lot of pairwise comparisons, for instance, if we compare stage 3 white to stage 3 purple, we'll get at genes that are involved in the difference between being white and being purple. We don't care about those genes, but we'll also get them in this comparison between stage 4 white and stage 4 purple. So any genes that are differential between here and here, we can exclude because they're about being white or purple. And you can do that with all of the pairwise comparisons, and if you do it, you end up saying that the only genes we're interested in, the ridge-inducing genes, are the ones that differentially express between stage 3 purple and stage 4 purple, and stage 4 white and stage 4 purple. Those are the 2 pairwise comparisons that give us ridge-inducing factors. So we did that experiment. The first thing you do when you get data back from this is a PCA to try and work out whether you've done it right. And so basically the 4 colour codes are the 4 samples, stage 3 purple, stage 3 white, and so on. And we've got 5 replicates of each one, and they cluster together within their colour coding and separately from each other. So that's great. That tells us we can actually differentiate those 4 tissue types by the genes that are expressed in them. What you then do is go have a look at the genes that are differentially expressed. And what won't be obvious if you have never done this and you've only ever looked at the headlines of papers that have, is that your bioinformatician spends a few weeks fiddling around on the computer and then comes back to you and says, there are about 8,500. Which one do you want? And you go, oh, that's not good news. So it's very much a needle in a haystack, trying to do this kind of differential transcriptome work. There are still lots of things that are differential, even though we have this careful four-way comparison thing. I'm not going to show you the 8,000. I'm going to talk a little bit more about how we try to understand how the ridges form, and then come back to one gene that's jumped out as really important at the end. So while we were doing that, we also started to think, well, could we imagine theoretically how these ridges are forming? And we discovered that actually there's a whole literature out there in the engineering field about how to make ridges on the surface of a material like this through mechanical buckling. So no genes, this is engineering. So the idea here is you've got two different materials, black and the stripy one. They're bound together at the surface, and if you shrink the top one, which in this case is done by cooling it, it shrinks, but it can't shrink because it's bound to the lower one. And if the things have the right property, what will happen is it will buckle. That's the energetically most efficient way of dealing with the forces that are applied to it. Now, we thought that was really interesting, except that, of course, when a flower develops and grows, it doesn't shrink. So how could we get the same effect? Well, actually, the engineers had an answer for that too. So I'm showing you a little movie here where we've got two materials bound together again, and we're dropping, that's a pipette tip, we're going to drop a drop of ethanol on the top one. There's the drop of ethanol. And because those two materials, again, have the same thing going on, the upper one wrinkles, all of those little ridges that have appeared is buckling because it's swelled up, but there's nowhere for it to go. So you don't have to shrink something to get this buckling. You can expand the top more than the bottom, and that has the same effect. And we thought, oh, that's interesting, because actually we have got expansion going on as our flower grows. So using this idea, we developed a theoretical mechanical model to try and explain the formation of these ridges without genes and just with mechanics. And the idea of this model was that there were only two parameters that mattered. The degree of stretch, this is our lambda term here, on the x and y axis of the petal cell was the first parameter, or set of parameters. And so the idea is that you can move from one pattern, that's this disordered pattern, to a ridged pattern, like this one, simply by changing the degree of stretch. And the second parameter is the amount of cuticle. So this is the overproduction of the cuticle on the top, which is generating that overgrowth in the same way as the drop of ethanol did on the two chemical materials I showed you a minute ago. So here's the same slide with the same stretches, but you can see if you change the beta term here, which is the amount of cuticle production, you get different regions having different patterns. So again, you could keep to exactly the same stretch terms. You could be sitting here at, say, 3 on the x-axis and 1 on the y-axis, but if you increase the cuticle production, you'll go from a smooth surface to a ridged surface. So it's a nice simple model, which is always a good place to start with a model, and we set out to test it. So we thought what we'll do, we'll modify the amount of cuticle, that's the beta term, and we'll modify the cell growth, that's the lambda terms, and we'll see if by changing those things, we change the pattern on the hibiscus surface, and that fits with the model. So to do that, we needed to develop transformation for the hibiscus. Again, I imagine this week you'll hear a lot of data from people presenting transgenic experiments with plants that are able to do interesting things. When it's not a model system, you have to develop that process for yourself. But the principles are quite simple. So you take, in our case, hypocotyls, you transfect them with an agrobacterium that contains the gene you're interested in, you make callus, so undifferentiated tissue here, like little tumour cells. And from those, if you apply the right hormones in the Petri dish, you can get a little shoot produced, which you can then chop off and stick in a bit of medium to root, and eventually you end up with a plant in a pot and then a bigger plant in the greenhouse in flower. And the whole process takes about 3 months. So using that, we're then using genetic modification to perturb the parameters of our model. And I said the first one we wanted to do was change the amount of cuticle the petals produce. So just to show you how we do that using one particular gene as an example, this is the SHINE3 gene. I told you when I told you how cuticle's made that the SHINE transcription factors activate the whole process. So these are the master regulators of how you make cuticle. So we cloned one out of hibiscus and we designed ourselves a set of experiments. So one set of transgenic lines in which we attach a constitutive activator to the gene. That should mean it produces masses of cuticle. One where we just overexpress it, a bit too much of the gene. That should make a bit more cuticle. Then we've got our normal plant. Here's one where we've made a hairpin loop which should down-regulate the gene, so you get a bit less of it, a bit less cuticle. And here's one where we've attached a constitutive repressor, and that should mimic a complete loss of function of the gene. It should give us lots less cuticle. And the lovely thing in here is that we already know what this gene does. We're not trying to figure that out. We just want to make variants which have different amounts of cuticle in them. So it doesn't really matter if some of them are better aligned than others. We're going to get a range here, and that's going to give us the data we need. So just to show you a couple of snapshots of data, we're looking here not at the front of the petal where those ridges are, but at the back where they're not. On the wild type flower, they're smooth. If we increase cuticle production with one of these overexpression lines, then we start to get wrinkling of the surface, which suggests that yes, increasing the amount of cuticle makes it more likely you'll wrinkle. Here's another version. We're now looking at the front where the ridges are. Here's your wild type petal. It's got quite a lot of cuticle. Here's a transgenic line where we've reduced the amount of cuticle and we've lost the ridges, so that fits as well with the model. So far so good. Changing the amount of cuticle, the beta term, does affect the pattern you get. What about modifying the stretches though, the lambda term? Well, to do that we just used a membrane-bound fluorescent marker so that we could measure the cells properly and we could track their growth. And we worked out our stretches, which are done very simply. It's basically the length in each axis before growth and after growth, and you calculate your stretch terms from that. And we did that for our wild-type hibiscus, and knowing what we know about its cuticle production, this is where it sits in the model. So that's good. It's in a region where it should have ridges, and it does. But it does mean that in order to change its pattern, we're going to have to move it right across this boundary, and that's a very long way to change a cell. So we, again, designed some transgenic approaches to do it, and this is our absolute best transgenic line for these experiments. We have managed to change the size of the cells from 100 microns in length to 40 microns in length. You can see they're no longer long, they're much shorter, but you can also see they still have ridges on them, and that flower is still iridescent. And when we put it into the model, that's because we haven't managed to move it across a boundary. It's sitting here on that model, and we haven't actually managed to make the difference. So this is an area where we're a bit stuck. We're struggling to actually change the growth of the cells enough to change those terms and check our model. We're currently using pharmacological approaches, so taking chemicals that change cell growth and applying them to this transgenic line to see if we can just shift it a bit further across that boundary, but not having any luck at the moment. But while we've been doing this, we've been looking at a lot of flowers down a microscope, and thinking about those flowers, we realised that actually maybe our model was just a bit too simple. So if you actually look at the petal in cross-section, you've got the upper epidermis where the ridges are, you can see them all here, picked out in red, and the lower epidermis where the ridges are not, it's smooth. But actually when we look at the cells on the upper and lower epidermis, they're the same size and shape, so the lambda term should be the same. And actually when we measure the cuticle production, it's the same too. So it almost disproves our model. The two surfaces have the same lambda and beta terms, but different morphologies. And at the same time as realising that, we realised that we had really missed something in this engineering literature, which is that for all of this to work, either with the movie I showed you or with the static picture here, the top layer has to be stiffer than the lower layer. And we thought, well, that can't be true, can it? Because our top layer is cuticle, which I think of as sort of like the treacle on the top of a treacle sponge pudding. It's kind of secreted, goopy stuff. And our lower layer is the cell wall, which is rigid and stiff and made of cellulose, and it's a box that the cell lives in. So it can't be the case that our top layer is stiffer than the lower layer, we thought. But we thought we'd better check it. And so to check it, we developed tools to measure the stiffness of the petal in layers using atomic force microscopy. So this is a microscopy technique where you basically indent gently a surface material, and a laser beam is deflected, and by how much it's deflected you can work out how stiff the material was that you tried to indent. And normally when people do AFM, they just indent the whole thing and they say that's the stiffness of the cell or the epidermis or the petal. What we wanted to do was indent the cuticle and the cell wall, the extracellular matrix, separately and get proper measurements of both of those. And we spent a lot of time trying to develop techniques for this. So it's hard not to emphasise how much of research is about developing the tools and techniques to try and figure out the things you're trying to figure out. We worked out how to peel the cuticle off. When we do that, we destroy it, so we can't measure it separately, but we can measure the cell wall without it. And by doing that, we worked out how far we had to measure, how far we had to indent in order to get to the cell wall. And we figured out that we could digest everything below the cuticle away, leave the cuticle floating in liquid and stretch it, and that would give us a measure of its stiffness, and we could compare that to what we got if we indented to different degrees to work out what the right depth to indent was. And again, to cut a long story short, the answer is that at stage 3, before the ridges develop, the cuticle is stiffer than the cell wall. That really surprised me. But the really big change happens as the petal grows. The cuticle gets a bit less stiff as it grows. The cell wall gets much, much, much less stiff. And of course, with hindsight, that's because as the cell elongates and grows, the cell wall has to become less stiff to allow that to happen. So it's in that long growth of the cell as it stretches that the cell wall relaxes and allows the buckling to occur, because the cuticle now becomes many degrees stiffer than the cell wall below it. So with all of those in mind, we have a new model. Sadly, where we had 2 parameters in the old model, we have lots now. We still have cell elongation because you need that to organise your ridges. We also actually have turgor-induced deformation of the cell, because if it's not flat enough, you won't get ridges. We have the amount of cuticle, so the thickness, that beta term from our original model is still important, but we also now have the stiffness of the cuticle and the stiffness of the cell wall, and those have to be at the right ratio to allow the thing to buckle. And so with this new model, with these 5 parameters in it, we're able to model and measure what's happening in our hibiscus. I'm showing you here models on the left of the whole of the cuticle, and on the right is a close-up of the top part of the left. And what's happening is that we fix the stiffness of the upper layer and the lower layer to those that match our hibiscus, and we're applying extra cuticle material at the rate at which it's applied to the hibiscus petal, and you see that what happens is that it buckles and it forms ridges that look very much like the ones we see on our flower. And we can even determine the wavelength, which of course affects the colour you produce, by controlling the degree of stiffness and the ratio of stiffness of the 2 materials. Okay, so it's all mechanics, except it's mechanics with a living tissue. So how do you determine the stiffness of your cuticle? Well, the answer to that is chemistry. I was rubbish at chemistry at school. And yet another thing I've had to learn about. The cutin polymer stiffness is determined by all sorts of things. It's determined by the fatty acids and how much they're cross-linked, and by other things, waxes and terpenoids and flavonoids that are in there. And it's all really, really complicated to measure. And the way people analyse cuticle chemistry is they take their tomato, they peel the cuticle off, and they mush the whole thing up. And we didn't want to do that because we wanted to know what the chemistry of the white bit of our petal was, and the purple bit, and the front and the back, and be able to compare them, because we wanted to look in a single petal. So with colleagues in the chemistry department at Cambridge, we developed this technique where we apply a droplet of solvent to the surface of our petal, and then suck it back up and apply it into a mass spec, and we get an answer about what's dissolved in that droplet of liquid. And we can do that on the white part and on the purple part and compare them, or on the front and the back and compare them. The answer is lots of things, lots and lots of things, all with very complicated names. But a good way of looking at the data is to compare these compounds and how frequently they come out in striated cuticle samples, so the purple ridged bit, those are the blue bars, and how frequently they come out in smooth cuticle samples, either the white bit or the back of the petal, and that's the orange bars. So you can see that there are lots of compounds which come out in them both, but are more prevalent in either the ridged or the smooth. But there are a couple of compounds, if you look closely, that only ever appear in the ridged cuticles. So those look really important to us. And this one here, which comes out in most of our ridged samples and none of our smooth samples, is a compound called dihydroxypalmitic acid. And this is a day when science is really good and you feel really happy about stuff. Because when I go back to that differential transcriptome I told you about right at the beginning, where we compared all the genes that were differentially expressed, and there were 8,500. The genes you're interested in are the genes that are differential between stage 3 purple and stage 4 purple, and between stage 4 purple and stage 4 white. And so this set here are the most exciting ones, and the most strongly expressed of those most exciting ones encodes the enzyme that makes dihydroxypalmitic acid, which was the chemical that was always found in the ridged samples on the last slide. So, I said it wasn't about genes, it was about mechanics. Mechanics is about chemistry, and chemistry is a product of genes, and so we're back at trying to do developmental genetics to understand how you make that ridged cuticle. And if we play with the genes encoding the enzymes that make the cuticle have the chemistry it has, we can do things like mess up the ridge pattern or completely take it away, and this lovely smooth one, which has a much less stiff cuticle, has also lost the blue iridescent shine on the petal. So we were able to sort of square the circle and put it all together. This is a single snapshot of data, and we're busy trying to work out all the components of the cuticle chemistry that give you the right degree of stiffness, and also the components of the cell wall that give you the right degree of stiffness too. So I've told you here a lot more of the development of this trait. Very briefly, its function and its evolution, and then we can all go to the bar. So the function. Does iridescence improve pollinator attraction? You'd like to think so. What's the point of it otherwise? But we needed to check that, because it's actually not like a CD. It's not even like the blue butterfly I showed you at the beginning. It's much fainter than that, or else somebody would have noticed it before us. So we designed an experiment to ask the bees whether it improves the speed with which they find flowers. So here's our flight arena. We've got 3 identical targets. These are blue discs of plastic, right, with a little cup of sugar on the top. And the experiment here is you let a bee in, you let her find the first disc and feed, and you time her from taking off to landing on the second, taking off from the second to landing on the third. And if you do that enough times for one bee, I can tell you how quickly she finds blue flowers. If I do it for enough bees, I can tell you how quickly bees find blue flowers. And if I repeat the whole thing with a diffraction grating on the surface of my blue discs, I can compare the two and tell you how much or little the diffraction grating, the iridescence, affects how quickly bees find flowers. All right? So here's the data with red flowers, not blue ones. The first bar is the flight time from the first flower to the second. Second bar is the flight time from the second flower to the third. It takes a bee 4 or 5 seconds to find a red flower in this experiment. If we add a diffraction grating to the surface, that drops to 3 seconds. Hooray! Iridescence makes it easier for the bee to find a flower. If I drop a red button on this carpet, it'll be hard to see. If it's iridescent, as I move my head looking for it, the colour change will catch my eye, and I think that's what's going on here. But I told you at the beginning about all these arguments I had with the physicists who said how rubbish the iridescence was and how not very good. So at this point we're a bit scared, because this experiment has been done by imprinting a CD on our red plastic disc. This is perfect iridescence. What's going to happen if I do it with a flower? If I make a replica of a petal and put that on the surface of the CD? We were really worried that we'd end up back up here and that it would turn out that we'd been studying something that was totally irrelevant. But actually, floral iridescence, so this is a replica of the surface of the tulip, increases foraging efficiency just as much as perfect iridescence, or maybe more, depending how you do the statistics. And I was able to smirk at my physicists and say, look, actually, the biology's just fine, thank you very much. It's perfectly good enough. Bees' eyes can only cope with a certain degree of iridescence, and the flowers are just good enough. And they went, okay, all of biology's rubbish then, not just flowers. But it gets better. We did a second experiment. This is an experiment that is about how easy it is for the bee to form a search image or remember a flower. So if you watch a bee foraging, she doesn't go buttercup, buttercup, bluebell. She sticks with one flower type for a foraging bout and then goes back to the hive. And they do that by forming what we call a search image, a picture in the mind of a rewarding flower and sticking to it. It's a sensible foraging strategy. So in this experiment, I've given the bee lots and lots of opportunities to forage on these blue flowers, and they've always got sugar in them. So she should form a really good search image of blue flowers. And then I put in place what we call distractors. They're quite similar in colour, but they're a bit different. They're purple. If she's formed a good search image of blue flowers, she should not go to these purple flowers. She doesn't know what they are. She's foraging from blue. If blue is hard to form a search image of, she'll go to the purple ones too. And again, I can do that whole experiment, and then I can repeat it again with a diffraction grating on top of these and see what effect the iridescence has on her ability to form a search image. So here's the data with a perfectly iridescent diffraction grating. It's presented out of 20. So when there's no iridescence, she gets it right 70% of the time, 14 times out of 20. So this is a hard experiment because those blue and purple flowers are quite similar. When I add a replica of a CD to the surface of these discs, she now gets it right 50% of the time. She's lost the ability to form a search image if I put a diffraction grating on top of the flower. That's not a good thing. But if I repeat it with floral iridescence, so here we are again, no iridescence, get it right 70% of the time. Floral iridescence, still get it right 70% of the time. Floral iridescence, unlike perfect iridescence, doesn't compromise search image formation. It's the perfect combination. So the iridescence that flowers produce is not a nasty bit of weak, wobbly biology that can't be as good as the CD. It's as good as perfect iridescence at increasing salience and foraging efficiency, and as good as no iridescence at ensuring that the bee can actually remember the flower. It's perfect. It's optimized for the eye of the receiver, which is a bee, not a physicist. So, I've shown you how you make them. You just have to have cuticle and cell wall and grow them in the right way and they buckle. And I've shown you that they're really good because they make it easier for bees to find flowers. So it's not that surprising that when we search across not 400,000 species, but the 8,000 I've got in my botanic garden, we find that iridescent flowers have evolved repeatedly, at least 10 times independently across the angiosperm phylogenetic tree. And so my job for the next few years, and thanks to a nice grant I got earlier this year, is to spend the next few years figuring out how it's evolved independently all of these different times. Does it work through the same process? Does the same model apply in these different systems? And does it work in the same way to the pollinator eye? So I'm going to really enjoy exploring that with a whole new range of flowers that I know almost nothing about and will have to learn about as I get going. It's one of the great things about a life in science, that you're always having to learn about things you know nothing about. Bees, cuticle, chemistry, and these flowers as well. So I'm going to finish just by saying, although I've shown you a whole range of curiosity-driven research, hopefully shown you that it's great fun and you get to just explore whole sets of different angles of things you knew nothing about. It does also have purpose and use. So the work we do, even though that's not where we start, often ends up informing crop breeding. The work I showed you, for instance, with nectar concentration and efficiency of grip and trade-off has fed into breeding programs on Faber bean here, which is commonly grown across the UK. Is nectar rewarding? And two of the varieties that were being commercially grown before we started this work had nectar with such a low sugar concentration that no bee would bother to open the flower to get at it. And we were able to tell the breeders and farmers about this work, and they stopped growing those varieties. So we want to make sure that the varieties that are grown have the best reward for the pollinator populations out there. We're also working in strawberry, and my long-term hope is that somebody will fund me to work on this, which is chocolate, which would be the most exciting plant in the world to work on crop production in. I'll leave it there. Thank you very much for listening. It's nice to see you all.